So my interest for biology and looking at organisms in the water stems back from when I was a young child. And I still remember I was about 4 and I had some of these animals in a small leaky aquarium I had at home, and I was looking at them and I was kind of blown away by the fact that these guys lived underwater and they were clearly breathing, I could tell, in a medium that would drown me or kill me. So my name is Martin Grosell. I'm a professor in marine biology at the Rosensteil School of Marine Atmospheric Sciences at the University of Miami. I work mainly on fish, but also on invertebrates. And what we look at mostly is the interactions between the environment and the organisms that live in that environment. As you're aware, combustion of fossil fuel have increased atmospheric CO2 immensely since the Industrial Revolution. That CO2 in the atmosphere collaborates with the oceans, and the elevation of CO2 in oceans causes acidification, which have had a lot of problems, mostly for calcifying organisms. You're aware of corals, for example, that suffer from lower pH conditions. We've looked mostly at fish, and fish have for a long time been assumed to not be affected by ocean acidification because they are very proficient acid-base balance regulators. The way fish maintain their internal pH when CO2 is increased in the water is that they retain bicarbonate. And they can do that in 2 ways, either by excreting acid or taking up bicarbonate from the water. The fish we've looked at seem to be relying on bicarbonate from the water. And one thing that's sort of gone unnoticed up until about a decade ago is that fish are actually calcifiers. They form calcium carbonate precipitates in the intestinal lumen. Elevated CO2 affects the production of this calcium carbonate. In fact, it increases the carbonate production in teleost fish in saltwater. And what's really interesting is that the carbonates produced by fish actually contribute to the inorganic carbon cycle by up to 45% of newly produced carbonate in surface waters. And what's going to happen with increased CO2 levels as we move forward in a climate changing environment is that fish are actually going to produce more and more carbonate and take a more and more important role in the inorganic carbon cycle, which is in contrast to what we know about marine algae that are calcifying and also coral reefs, for example. So I became aware of the Gulf of Mexico Research Initiative, which was money made available for research into the impact of oil in the Gulf of Mexico. They allowed for more independent and free study of impacts, and that's what we've been doing over the last 6 years in a large research consortium called RECOVER. And in that consortium, we've looked at sort of sublethal effects on animals exposed to oil, mostly lab-based studies, and we've tried to connect that to what happens to populations and the ecosystem in the northern Gulf of Mexico. And I will say the Deepwater Horizon oil spill, probably the biggest environmental disaster we've had in North America. 5 million barrels of oil spilled into the northern Gulf of Mexico. There's very little good about that. The one thing I will say that's come out of it, though, is a lot of knowledge about the northern Gulf of Mexico and the organisms that reside in that environment. It's long been known that oil exposure during embryonic development in fish will cause cardiovascular problems. So it leads to fluid accumulation around the heart in the yolk sac area. We call that pericardial edema. What's also often seen is other malformations such as spinal curvature, reduced eye development, reduced jaw formation, and essentially these larvae that hatch out after being exposed during embryonic development are incompetent. They will not survive past first feeding. And in fact, eye formation and eye development is impacted severely by oil exposure. And actually that occurs at lower concentrations and earlier during development than the cardiovascular effects. In general, for toxicity, you expect the smaller organisms to be more sensitive, and that's also the case for oil. But what we've shown is that even later life stages of fish, so larger animals, including adults, are surprisingly sensitive to oil. We know that the stroke volume, so this is the amount of blood leaving the heart for every contraction, is greatly reduced in adults. And that means that they are less able to swim, they're less able to take up oxygen, and therefore cannot migrate, capture prey, avoid predators to the same extent as unexposed individuals. We've then looked at that at the cellular level, and we've shown that isolated myocytes, so muscle cells from the heart, they are less able to contract when the fish has been oil exposed compared to non-exposed. This microscope here has a little chamber down here where we can keep individual cells, and then it has the ability to perfuse the cells with different solutions. And then when it's doing that, it's recording the cells, and we can then, through these wires here, stimulate them electrically. So this is what happens in your heart. There's, the nervous system will give your heart a shock every so often. Right? As a scientist and as a professor here at the University of Miami, I am blessed with a work environment and a work situation where I enjoy and look forward to going to work every day. And that's because I do what I care about, and you really cannot put a price on that. Actually, the favorite thing about my job is interacting with young students. Sure, I learn from my research, but I actually learn as much from the students and postdocs that I work with, and that seeing them develop into independent scientists is perhaps the most satisfying part of what I do.